Hello and welcome to this webinar titled Next Generation Processes, What Model Works the Best in Asia, hosted by Biopharma Asia magazine and presented by Ravishankar Kasturi, Vice President, Head of Manufacturing and Process Development, Reliance Life Sciences, and Britta Manser, Scientist of Continuous Bioprocessing, PAL Biotech. My name is Stephen Edwards and I will be your moderator. Now, please allow me to introduce our first presenter, Ravishankar Kasturi, Vice President, Head of Manufacturing and Process Development, Reliance Life Sciences. Ravishankar Kasturi is Vice President, Manufacturing Operations and Process Development at Reliance Life Sciences in Navi, Mumbai, India. He manages all aspects of process development and GMP manufacturing to produce biosimilars for clinical and commercial supplies. The portfolio of products include proteins derived from microbial fermentation, mammalian cell culture covering cytokines, monoclonal antibodies, hormones, and glycoproteins. His prior assignments were at Dr. Reddy's Biologics, Hyderabad, and Unisanko Limited, Hyderabad. He receives his MTech in Biochemical Engineering from Andhra University, Visakhapatnam, and his Bachelor's in Pharmacy from Gulbarga University, India. Welcome, Ravishankar.
Thank you, Stephen. All right. Good morning, good afternoon, and good evening all. I would like to thank you for taking your time to join this webinar. As Stephen did the introduction of mine, I'm going to talk about the presentation titled Next Generation Processes, What Model Works Best in Asia. I'm going to talk about the various processes which are currently available, which can be adapted in the manufacturing process. Okay. I'm going into the presentation mode. Yes, I hope you all see the slide set. Let me go to the contents of my presentation. So now I'm going to talk about, in my presentation, I'll briefly talk about what are biosimilars, even though it's biosimilars, everyone knows what biosimilars are, and I'll talk what's the need of the hour, which is the affordable therapies, and how biosimilars contribute to the affordable therapies. And I'll also talk about the various opportunities we have for biosimilars in the emerging markets, Asia being one of the emerging markets. And opportunities also has its own set of challenges. We'll also look at some of the challenges that we currently have. And I'll talk about the methodologies, the various the ways of doing the manufacturing and having a technology set up. And at the last, I will conclude my presentation with a conclusion, making a summary of the talk that I gave. So I'm going into the slide number 4. So the affordable therapies, that's the need of the hour. We all agree, and it is also very important to have affordable therapies for various regions. We have seen that in pharmaceuticals, how the generics have come in, and that's being the same is being seen in the biopharmaceuticals with the biosimilars coming more and more, getting acceptance of biosimilars in the various markets. But going back to the affordable therapies, the Asian markets are widely recognized. One of the most important growth frontiers for the drug development. As I said, we already seen that in pharmaceuticals, and now we are seeing this in biologics and the other precision medicines like gene therapies and other cell-based therapies. Why is that? Because we have 4 billion population, and with Asia Pacific, that's also viewed as the growth engine for the global biopharmaceutical industry. In that, and it is a competitive age, and there is competition in each and everything that we do. So how do you make a product and how do you win the product, that's where we need to bring in the speed, agility, and innovation in whatever we are doing it, so that the product that we develop and deliver and the process, including the process that we use to develop that product. So all that becomes a part of affordable therapy that anyone try to develop that. So it has a very huge impact, enormous potential for the companies that they need to choose the right path. Going into the slide number 5, that slide I detail the various opportunities that we have currently in biologic space. And it is widely known that many of the drugs are becoming off patent. If you look at the top 8 biological molecules which are listed on the slide here, if you look at the freedom to operate, and in both US and Europe, most of the drugs are already off patented, and some of them are very near they go off patent, so that most of the companies who are in the development path have freedom to develop the product and market the product. And this is a total list of 8 products which are the top. I listed the top selling products. But what it clearly says is several biologics, biopharmaceuticals are going off patent, and it is a tremendous scope for the companies to develop and take them into the market. The opportunity starts with due to the high cost of the branded biologics, and also in countries like China, Brazil, Russia, they are trying to be independent. Like right now, if you look at the availability of biologics in the space, they either have to import the molecules from the countries outside in. But that's the area where they're also trying and getting into the manufacturing and supplies from themselves. So that's also giving opportunity for those markets to build the domestic biologics. And going back to the Asian context, the need for biosimilars in Asia is also great, and again, the Asian markets are going to influence faster because knowing the worldwide adaptation of biosimilars across the world. So if you look at the IMS Health and drug sales data in the emerging markets, the markets I talked about, it's almost as twice as the global rate. So that talks about the significance of the manufacturers in the emerging markets and this growth potential they have with the biosimilars. Okay, I'm going into the slide number 8. I'll talk about the regulations. We see the regulations are also evolving. Not all countries have the same type of regulations. If you see from the guidance point of view, we already have guidance detailed by EMA. We have from FDA, US FDA, and WHO, which have set the general principles to how to demonstrate biosimilarity in terms of the quality, safety, and efficacy in that. But if you look from the various other regulations point of view, they are wide, they are varying, not all are same. So we don't find one single regulations which we will be seen across, accepted by all various nature. So that it's very important to understand that what are those regulations, and whenever we are targeting those particular markets. If you look at some of the regulations in India, we have revised the regulations in 2016, and we also if I look at Singapore, Malaysia, and South Korea, they are very, very, very similar to what EMA requirement is. And some of the countries there, we do also have some countries who they have their own regulations, and sometimes they look even a biosimilar molecule as a new biological product. That's the way their FDA looks at it. So it's very important to understand the regulatory requirement to ensure that product remains in compliance from the time we start developing till the time we push the molecule into the market. That's about the regulations. Let me go to the next slide, slide number 9. So now let's see, how do we win this race? So winning is, of course, that's the requirement, that's what we intend to develop the product. So how do we win? First, we need to choose the right product and the therapeutic area. So that's the choice that we need to make. We need to do our own market research to come up with what's the product that we need to develop this. And also we need to have pockets of non-consumption. Means if there are pockets of non-consumption, it's important to understand those so that we strategically put our product there so that it gets available in terms of the reach of the product available for those markets. And it's also important to partner with the local players so that we understand the local requirement and the knowledge that's required. Many regulations insist that the product that we intend to sell in their area needs to be manufactured in that particular location. So it's important for us to also have a collaborations in the local market so that we can push the product into those particular markets. Right. Let's see. Cost of the goods is again a buzzword in manufacturing. Being development or in manufacturing, it's the buzzword, because why is it important now? When we start investing it, so it becomes very important. We need to understand when to invest the product, where to invest, how much to invest. If we invest in too small of capacity, sometimes we may face out all the stock out options because we're not able to make as much as market needs. If you invest in too much, it adds to the overheads and the cost of the goods go up. So we also need to take a balanced view on should we do it all in-house or should we go to a CMO and take their help in developing the product, also manufacturing the product, and what's the right technology platform. So that's where I'm going to spend some more next 10 minutes about the various technology platforms that we have. Either the stainless steel we should be using or single-use systems, it should be do a batch process, perfusion or continuous. So these are the various questions one needs to put in and work on when we are working towards the optimization of the cost. There will be challenges, and there are challenges. One is funding at the developmental stage, and also the other challenge is the scaling. I talked about in my previous slide about the scale of manufacturing. Many times our calculations go wrong on the scale. We assume the requirement at a particular scale, we do everything at that particular scale, and suddenly sometimes the scale looks too big and the product goes in the inventory, or sometimes it is too small so that we have to scale up and then do a lot of rework and validation associated with that. So that's a challenge that we need to understand, and meeting regulatory obligations is again a challenge because of various requirements coming from various areas. And also now, if you look at what's happening across the world, a lot of innovative companies like the big pharma companies are also coming into the biosimilars. That also gives a big challenge and competition to the small biosimilar manufacturers developing the molecules. Let's see what is the right technology platform. If you put the questions, what should be the right technology platform? It should be robust, and in my opinion, it should be robust and manufacturable. When you say the robust, so that if you have multiple products, again and again, we should be able to use it for manufacturing purpose. Consistent, the product that you are making should be consistent, it should be competitive in terms of the cost of the goods, and as long as you have a standard platform, we should be able to make the changes both in the product and also in the process. And that's the reason, that's from that point of view, it is important to have the right technology platform. And again, whether we should look at the legacy platforms or new platforms, that's also something that we should look at. If we look at the process methodologies, there's several in the market, but if we want to simplify, one, stainless steel stands on the top, which says it's a historical, it's a default technology which has been there for several years, decades now, and which has been still as a low risk if someone wants to use it. Then comes the disposable technologies, because it brought in a lot of flexibility, capability, capacity. Perfusion technologies, there are many in this space. We have alternating tangential flow, call it as ATF. Internal spin filters, again, this is one which exists several years old. centrifugation, cell-settling devices. There are many more which are coming up in this space. Hybrid technologies, someone who are very don't want to spend a lot of money, be competitive, worried about the cost of the goods. Many are going into the hybrid technologies where they use single use on the upstream and then they put the entire downstream in a conventional way of purification. Then we also have a fully continuous system, wherein it's a technology that is coming up. Everyone is trying to understand. I would say it's an early stage of development for the large biotech companies. So even though some of the small biotech companies already started implementing using it. So here, what important is accessing the choice of the best fit is very important. Now on slide number 14. If you look at the difference between a stainless steel versus the single use, it has already been I think there are several conferences which talk about what are stainless steel, what's about a single use, what's advantage we have. But still, SS sometimes most competitive, cost effective. For the low titers, if you have a very low titers, and that's the reason why we want to make manufacturing at a very large scale. That's the case. Stainless steel is the best option, but most of the times it's not the case now for the manufacturers. Single use still becomes more competitive as becomes tight as the titer goes up, because as the titer goes up, we don't need larger volume. The entire volume handling goes down, and cost of goods is one of the consideration that we should bring, but we also look at the flexibility, throughput, and time that we take for a facility to come up and take the product into the market, and regionalization, how easy that we can have the plant set up. All that has advantage with the single-use systems. But now, if you look at the market, single-use already, most of the manufacturers embrace the single-use technology because it has reduced the reduction in the capital investment. Why low capital investment? Because low capital translates to lower fixed costs and less financial risk. That's the reason why single-use has its own advantages. I talked about the timeline for implementation, which is much less with single-use systems. And also the flexible, we don't do CIP, SIP, you saves enormous amount of water, and also cost effective for low volume biosimilar products. If you have a very low volume product, we don't need very large 20 KL fermenters to manufacture these type of products. And most of the companies, as I said earlier, already single-use systems become a mainstream way of manufacturing. Now, I will introduce to this new concept called continuous processing. By definition, any unit operation is continuous if it is capable of processing a continuous flow input for a prolonged period of time. That's my definition of what continuous is. If you look at the history, this has been adapted in many other industries. Like if you look at the automobile industry, it has been already adapted there and widely used in. But it has been wherever there is a capital-intensive sectors, this technology has been adapted. If you ask, is Biologics, Biosimilars capital-intensive? Definitely yes, it is very capital-intensive. But in terms of when it comes to the adaptation of the technology and the validation of adaptation of the continuous processing, it has been a bit slow because of some of the challenges. I'll go in my coming slide, I'll talk about what those challenges are. Let me talk about the strategies for continuous manufacturing. I'm on slide number 17. So if you look at the various options we have in upstream, we have continuous perfusion reactors, again, being there for decades. In clarification systems, we have depth filters, acoustic wave separators, which are there, which are coming up now. Downstream, multi-column chromatographies, which are connected in series, online buffer preparation systems, and single pass transition flow, integrated continuous, and a way of integrating all of this are some of the strategies which are now being available in the continuous manufacturing. Slide number 18. Let's look at some of these. What's the advantage that this continuous system will bring in? One is, of course, the cost. Because of reduced cost, what it does is use of smaller equipment, and smaller equipment generally cost less and they require smaller facilities. So the entire cost of manufacturing, of running the facility is much less with the continuous bioprocessing. Productivity, if you are using in a continuous way, since they are operated continuously, we need little space for storage vessels, a lot of other buffers that are required in the manufacturing process. And we can expect higher yield. The overall FTE's full-time employees requirement might be slightly lower compared to the conventional process. Quality, of course, depends on the product and the process. We can have, if you have a product which is a bit challenging in terms of the quality as it gets degraded with time, continuous is a way that you have a hold on what the quality of product that you are making. And of course, the protein degradation. Of course, it also brings in the flexibility because of more adaptability and facility utilization, and also if you want to replicate the facilities elsewhere, I think that's the way it's going to be in. And there are a lot of other advances which is also happening in other areas in terms of the computing technologies and mathematical modeling. All this actually facilitated in adaptation of this continuous manufacturing. Okay, I'm in slide number 19. Let's look at, I think I skipped one slide. Let me go back. Okay. No. Okay. Sorry. I'm on slide number 19. Let's look at adaptation of this particular technology in the biosimilars and the biologics, especially, be it a biosimilar or an innovative molecule. If we look at the adaptation, interest is on rise. That I know I'm seeing from my own organization point of view, and over years, FDA has many times vocal in giving support for this continuous manufacturing, which also gives confidence for the manufacturers in embracing this technology. And it also opened up a lot of options for making continuous manufacturing feasible. In that event, I read one of the report of Biopharma Associate, which says that 10 products of currently products which are being manufactured are through either perfusion or continuous downstream processing. So 10% is good to start, and I'm sure in the coming years it's going to go up. So one thing I could definitely say is the adaptation of the continuous bioprocessing will very likely to follow the trajectory that had happened to the single-use systems. Now, single-use systems is there in every facility that we go in, and likewise, as we move forward, I think we're going to see the similar adaptation of the continuous technology in almost all the manufacturers. This is my last slide. In my conclusion, I'm going to say the markets for biosimilars are growing very exponential. We have seen that not only in Asia, outside in every emerging market also. And it's very important to have smart technologies. That's the key for success of the biosimilars in these markets. And from the Asian market point of view, Asian markets offer huge potential for local and the global pharmaceutical companies which are developing these innovative treatments. And the companies will be successful only if they strip away the cost and deliver the value-driven product at a price that the market requires or the market demands. And similar to single-use systems, the continuous bioprocessing in near term will see a very increasing adaptation in that. That's all I wanted to say and talk to you about this various technologies, various technological solutions which are available for all the manufacturers who are in the biologics space. In that, I'd like to thank you all. Thank you for taking your time. And as mentioned by Stephen, I'm here. I'm on the call. I can take your questions once I think we finish Britta's presentation. Again, thank you so much.
Thank you, Ravishankar. Once again, I'd like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Britta Manser, Scientist in Continuous Processing, PAL Biotech. Britta Manser moved to PAL International in 2016 after obtaining a master's degree in pharmaceutical biotechnology from Zurich University of Applied Sciences. In her role as continuous bioprocessing scientist, she is focusing on the development and implementation and integration of continuous technologies, especially continuous chromatography applications. In the past years, she has implemented continuous bioprocessing solutions at various biopharmaceutical companies throughout Europe. I will now be handing over to Britta Manser. Welcome, Britta.
Thank you very much for the introduction, Stephen. So in this second part of the webinar, I'm going more into the details of continuous bioprocessing and showing you how continuous technologies are used for biopharmaceuticals today. So to pick up from Ravishankar's talk, we have seen a movement towards higher product titers in the past years, which goes along with a general batch size reduction. In addition, the biopharmaceutical industry, which has so far rather focused on quality and safety of the product, is today seeing a much stronger cost pressure, especially if we're talking about competitive markets such as biosimilars. Also, next generation processing involves the use of more multi-product facilities, which requires a higher flexibility in manufacturing. Now, solutions for such flexible but at the same time cost-efficient productions can be found in both single-use technologies and the newer continuous technologies. Some experiments that were made by early adopters in the biopharm industry, but also experiences from other industries where continuous is a reality for a longer amount of time, for example, in production of small molecule or in the chemical industry. From there, we do know that continuous production can lead to significant improvements, both in process economics and in process control. On one hand, continuous allows us to utilize the machinery or the equipment much more frequently, which means up to 24 hours a day the machinery could be running. And on the other hand, continuous production reduces the overall size of all our unit operation, and this has a significant impact on operating expenses. In addition, continuous processes are typically highly automated, typically more than what we are known from batch processing today. And this automation facilitates the process control, and that has a positive impact on both process safety, but also product quality. So overall, having all these points in mind, we now see that continuous processing is one of the most promising tools for process intensification, especially when we're talking about downstream processing. So moving to the next slide, talking of continuous processes, what we envision is a layout as you can see in this slide here. On the top, you see a fed batch bioreactor, which is feeding into a batch downstream process. And in batch, as we know it today, one step is happening after the other. In comparison to this, you can find a continuous downstream process below. Here we also have a fed batch upstream process, but it's leading into an integrated continuous downstream platform. After cell harvest, we're using the exact same process steps, including, for example, same filtration media, same chromatography resin, identical buffer systems. However, the unit operations are interconnected, and this allows one integral product flow throughout the whole chain of unit operations. This utilizes the unit operations more efficiently and at the same time avoids long hold times in between the processing steps. The unit operations also, typically, they can be downsized because now the product flow rates are generally lower. And this scale down, of course, translates into cost saving, especially if we imagine, for example, high-price chromatography resin, which now can be scaled down in the column size. However, to run such a process, we require technologies which are capable of dealing with constant flow conditions. So it's got to be technologies that can tolerate a continuous inflow, but also deliver a more or less continuous outflow of product. Certain technologies that we know from batch processing today, they can already handle such conditions, thinking of, for example, a filtration step or a flow-through chromatography. So these operating steps can be directly translated into a continuous process. For all other steps, however, we do require technologies which are specifically developed for continuous processing. And in this context, Paul has developed technologies in 3 core areas of downstream processing. So I'm now moving forward to the next slide. You can see here the overview of the 3 core aspects where Paul has created the new technologies. In the clarification segment, we work with acoustic separations. There, cells are separated from the supernatant using newer 3-dimensional acoustic waves. Benefit of the acoustic separation is that it doesn't block, so it can be easily used over longer periods of time, for example, in a continuous setup. In the next area, which is the purification, we can count on the Cadence Biosimilars system, which is a multi-column chromatography system. It allows us using up to 8 columns for continuous chromatography. You've already seen a schematic of such a multi-column chromatography in Ravishankar's talk. Now, in antibody purification, a low pH virus inactivation typically follows the capture chromatography. And here we work with the Cadence virus inactivation system, so that's the system on the right-hand side in the purification section. And this system allows us a continuous low pH inactivation of the elevate coming from the Biosimilars chromatography system. In the last section, which is the formulation, the batch process typically relies on TFF systems. So they are used for concentration and diafiltration steps. However, those TFF systems normally use a recirculation loop, and you can imagine this is not compatible with the continuous flow that we need in our processing. Therefore, we're applying single-pass TFF technologies, both for concentration and diafiltration. So with this portfolio, we have the full range of products that we need for PD scale continuous antibody platform setup. And when we look at the GMP-compatible large-scale version of all these technologies, the majority is available already, and the rest they are being developed as we speak. So there is a whole platform of different products which allows us to move forward with continuous processing. In the next slide, I'd like to show you how these technologies can be implemented, can be hooked all together to make one continuous antibody platform. So this picture that you see here is from the Research and Development Laboratory at Paul Biotech as it's set up in Westboro, Massachusetts in the US. This is a 36 square meter lab, and it holds all processing steps from primary clarification down to the final filtration. The process starts on the left-hand side with the acoustic separation device. Here, the cells are being separated from the supernatant, and that supernatant then further moves through some filtration and pre-concentration. In the next step, a multi-column protein A capture chromatography is performed. This is performed on the Biosimilars PD system. Right behind the system, the elevate is continuously inactivated using a low pH solution. And then when we move further downstream, the product is polished through a flow-through anion exchange and a mixed-mode cation exchange chromatography, both implemented on the second Biosimilars system. Finally, the product moves to the right-hand side of the laboratory, where virus filtration, then concentration, diafiltration, and final filtration are all performed. Altogether, this laboratory contains 14 to 16 unit operations, which can produce up to 6 grams of purified antibody per hour. So when you step into this laboratory, you'll find that all of the unit operations are constantly running all at the same time in order to produce our product. This platform has been tested under multiple circumstances, for example, with different feed solutions, and I'd like to share some of the results with you here. In the figures on the right-hand side, you can see the host cell protein and aggregate content of the product as it moves through the different unit operations. This assessment was made for bioreactor volumes of both 25 and 50 liters and for product titers of 1 and 4 grams per liter. And we can see that the platform actually well adapts to volume and titer changes. Overall, it was possible here to achieve productivities of over 120 grams per liter a day with an overall platform yield of roughly 56%. These results, so what we see here, they provide proof of concept for continuous antibody manufacturing. What I'd like to show you here in the next slide is that through implementation of continuous technology, chromatography resin and buffer consumption can be significantly reduced. So these are results coming from the same platform that you've just seen, and it's a direct comparison of a 25-liter bioreactor, which is on one hand processed with a batch downstream process, so that's the left column, and the right column shows you the same bioreactor, but clarified through a continuous downstream process. And what you can see here is the resin or the sorbent consumption, which is required for the proteinase step. So that was used with the Kaneka-Kanka-Bay proteinase sorbents. Then the Mustang Q, this is our anion exchange membrane, and the CMM hypercell is the mixed mode cation exchange chromatography sorbents. And you can see that through all three steps, the consumption can be significantly reduced. Going along with smaller column volumes, also the buffer consumption comes down in this platform by somewhat over 40%. So these are very promising results that show the proof of concept of continuous bioprocessing, and I'd like to now get the opportunity to show you some further examples from the industry. In this slide here, you can see the integrated antibody platform that Merck in Kenilworth in the US has set up. Merck is heavily driving their manufacturing of the future and has established some end-to-end, fully integrated, continuous processes for antibodies. And this picture here shows you the full platform. It starts on the left-hand side with a perfusion bioreactor, and then the product moves through several unit operations, including 3 chromo steps. The platform is fully automated with Delta V and has been operated in long campaigns of 3 months or even more. The second example I'm showing you here is the integrated continuous antibody platform from Bayer. This is in Leverkusen in Germany. It's a project called Moby Dick, and their continuous setup also starts with a perfusion reactor, which then leads to multiple steps of purification all the way down to drug substance. Bayer has also automated their process using Siemens PCS7, and they achieve outputs of roughly 25 grams of antibody per hour. The 3 examples that you've just seen now are all fully integrated continuous processes. Thereby, all the downstream processing unit operations are connected, and even upstream can be added to the chain of unit operations. Some early adopters, including Bayer, Merck, or MedImmune, have successfully created such processes already. This concept is making use of the benefits of continuous processing very intensively, but it's also the most advanced setting, as you can imagine. Therefore, what we see most often in the market today are hybrid processing scenarios. Hybrid processing is shown in the next slide here, and it is a mix of batch and continuous technologies with the goal to best meet the production needs. So what we do in hybrid processing is only a fraction of the process gets translated to continuous. So we start off with a batch process, and we look out for any bottlenecks or very cost-intensive steps. Typically for antibody process, you can imagine that would be a protein A capture step, for example, maybe followed by the pH virus inactivation and probably a second chromatography step. So we are looking for those steps which are most painful, let's say, in our production platform. And we translate these steps into continuous in order to make them more sustainable, more efficient for our processing. However, all the rest of the process remains in batch mode. And the experience that we've obtained in different case studies and work with customers so far shows that actually hybrid processing is a very attractive scenario for biopharm as well. I would now like to move into some more case studies, giving you a bit more details on the hybrid and integrated continuous process. What you can see on this slide here would be an example of a hybrid process, for example, because we are specifically looking at the protein A chromatography step. This data you see here was generated and published in cooperation with Sanofi Aventis in Frankfurt in Germany. Sanofi Aventis has tested the impact of continuous chromatography on the protein A capture step of their antibody process. Thereby, Sanofi has performed reference studies with their batch process first, so to see what the baseline process looks like in batch currently, and then they've translated this process onto the small-scale, process development scale continuous chromatography and from there performed a scale-up on the GMP-compatible large-scale chromatography system. All of this was used or was performed using the BioSMB system from Pol Biotech. The study here was carried out for 3 different operating points. That means that 3 different binding capacities and residence times were tested just to have 3 scenarios available for the protein A chromatography. The results that you can see here shows that all continuous settings in this study could give 4 to 5 times higher specific productivities when compared to the batch reference process. So that means that they could produce more gram of product per liter of resin per hour of processing time. In addition to this, the product quality attributes were assessed. So if you have a close look at the graphics on the right-hand side, we can see that host cell protein removal, DNA, residual protein A content, and product purity are very similar when we compare continuous to batch processing. So this is giving us already a good proof of concept, some good idea of how a continuous chromatography process can first be translated from batch to continuous, but then also scaled up to under GMP processing conditions. So this would be just one chromatography step. I would now go back to the idea of having fully integrated continuous downstream processing. So when we consider integrating downstream steps and hooking them up with the perfusion reactor, which is upstream, we have to consider the bioburden aspect. In perfusion-based processes, where an integrated platform runs for maybe 30 days or longer, bioburden control in the downstream steps becomes essential as well. Since everything is interconnected, some contamination somewhere in the downstream steps could easily spread throughout the platform. So now processing steps that have historically not been operated under strictly bioburden controlled conditions, for example, chromatography, now need to be upgraded accordingly. And Pol has therefore developed a continuous chromatography system. It's a single-use closed system which is specifically designed for continuous chromatography operations over longer periods of time, saying if you want to hook up your chrome system to the perfusion upstream reactor, for example. Several assessments were performed to test whether this continuous chromatography system is actually suitable for operating under these bioburden controlled conditions. So in the first assessment, the capability of the system to be installed under zero bioburden conditions was tested. This was done by installing the manifold, all single use, and then having a media hold test post-installation. Furthermore, it was tested if the system can operate under closed conditions over an extended period of time. So this was also performed with some growth media, which has been constantly recirculated in the BioSMB manifold. In both of these preliminary media tests, no bioburden was detected, which confirms that the system has the potential to be operated under strictly bioburden controlled conditions. In the last step of the testing, a chromatography step, being a protein A chromatography, was performed over the course of 10 days together with Sanofi Aventis. And the aim of this study was to verify the bioburden stability of the and process consistency, but under real process conditions. And in the next slide, I'm showing you the results of this assessment. The results are shown here. So we were using a 3-column process with some gamma irradiated protein A columns operated over the 10 days. Now, 2 of the 3 columns, they would always be in the load step. So they would consistently be loaded with the product coming from the perfusion reactor. And the 3rd column is at the same time moving through some wash, elution, and regeneration steps. The figure that you see here, that shows the overlay of the UV signals in the elution outlet. So every time one of the columns is in elution, you will see one of the UV spikes coming up. And what we see here is that there's a high process consistency because we can't observe any drifts in the elution profiles over the 10-day operation. So that gives us the time consistency. And also the 3 peaks are very comparable, which gives us a good column-to-column consistency as well. So in the end, we're having a very stable process over time. What was also assessed was yield, impurity removal, and product quality. And here we can also see very consistent results. In addition, what's very important as well, no bioburden was detected, which again confirms the proof of concept that this closed single-use chromatography is suitable for operating over extended amount of time, especially under GMP conditions. With this, I'm closing the case studies here, and I would now like to spend some time as well on regulatory aspects, as Ravishankar has already started in his presentation. So with biopharmaceutical manufacturers moving towards the implementation of continuous processing under GMP conditions, the regulatory aspects gain more importance. We know that both the FDA and the European Medicines Agency, they recognize the benefits of continuous processing, and Paul has also actively engaged with regulatory authorities in multiple geographies to, on one hand, receive their feedback, but also to receive their feedback on validation strategies, but also to educate them a little bit on continuous bioprocessing, since this is still quite new to all of us. So in these discussions, we have seen that there is a high level of engagement and enthusiasm of the regulators. They show to be very open and willing to learn about the new technologies. However, there are of course also questions raised when it comes to continuous processing, and these need to be addressed. 4 of the main topics that we are asked include, for example, the management of bioburden. It includes how you would define a lot in continuous bioprocessing, how to control any process deviations when you have interlinked unit operations, or also how you would define the design space using the quality by design approach, or how to address virus safety. Paul has developed and shared various strategies in different occasions that address the listed concerns and different regulatory topics in this context. Now, in this presentation, I'd just like to give you a brief overview of 2 of the aspects, as talking about all of them would just be too much for this presentation. But let's talk about the bioburden aspect once more. Concerning bioburden management, you've already seen how chromatography can, for example, be operated under controlled conditions. In addition, we here recommend using the exact same principle as is applied in batch processing today. So it literally comes down to prevention of bioburden in the process. This means that in continuous processing, we use closed single-use systems, and whenever we have to connect these single-use systems, we will use welding technologies or aseptic connectors. There's also the possibility to implement some strategically positioned bioburden filters that all helps us to maintain bioburden controlled conditions. So you can see there's different aspects that will help us to get the bioburden aspect in place. Another question I'm addressing, what we are asked very often is, how do you define a lot in a continuous process? Even though the process is running for days and weeks, you still have to define your certain batch. And here we have some information from the FDA. Already back in 2014, the FDA has published that they would use the same lot definition for continuous processes as they are applying on batch processes today. That means that as long as it can be shown that a product is of uniform character, a batch can be defined based on time, for example. So we could say, even though the process is running for several days, every 24 hours, that would be a batch. It could be also based on volume, so you could link a certain amount of end product to be a batch, or you can base it on the raw materials. For example, if there are several bioreactors leading into the same continuous downstream line, the batch could be linked to the raw material of the bioreactor. So you can see that the regulatory authorities have given a lot of room for the definitions here. Now, recommendations, like I've just shared a few, are available in much more detail and for more questions, and we're always happy to share them with you in separate discussions. So to sum up, I'd like to show you a picture here of a typical life cycle assessment. And you can see that already back in 2015, when the graphic was created, continuous technologies have already moved to the second tier, and we've recognized that the technology is rapidly moving towards general acceptance. The first biopharmaceuticals produced with continuous technology are expected to be filed very soon. In conclusion, we see continuous processing as one of the most promising tools for process intensification in our industry, especially when we talk about downstream processing. Various case studies with our end users have shown the benefits of continuous processing. including improvement in process economics, but also process control. The technologies to implement such processes are available today at PD scale, so for process development, and the majority of the process steps can already be translated under GMP large-scale process scale already. Regulatory challenges are currently being addressed, and we're expecting the first submissions of biopharmaceuticals involving elements of continuous downstream processes to be filed very soon. So it's an excitement field or area to be working in at the moment. So with this, I'm summing up. Thank you very much for your attention. And Stephen, I'd now like to hand it back to you.
Thank you, Britta. Now we'll begin the question and answer segment of the webinar. Once again, I would like to remind our audience that you can still send your questions in via the questions tab on the right-hand side of your screen. Now, our first question is, what are the prospects of biosimilar manufacturers in Asia?
Yeah, Stephen, I'll take that. Question is about what's the prospect of biosimilar manufacturing in Asia. So I will again go back to my presentation where I talked about the high price of the branded biologics and not being tough to reach many of the patients across the world. Because of that, the birth of biosimilars and biosimilars are being endorsed now everywhere, starting from underdeveloped to the developed countries and the markets. So if you look at the prospects, if you ask the prospects, and if we look at the disease patterns that we are having, the use of biological drugs, and the population, the huge population that we have, the health awareness that is getting created, and the economic growth that we are seeing in the developed markets and the emerging markets, and the sensitivity of the price and the regulations, and allotment of the budget, more and more budget is being allotted. From that point of view, all the biologics and the biosimilar space in Asia play a key role, and Asia is becoming a big hub for biosimilars. I could say that.
Thank you. Our next question is, how can a batch process be translated into a continuous?
All right, I'm taking this one. So typically, the batch or the continuous process design is always based on a reference batch process. We use that as a baseline, and then we create a design space for which we then evaluate the continuous process. So, for example, for the continuous chromatography, we perform some breakthrough curve studies up front. This will tell us the certain operating range and the design space that the continuous process can be designed in. And from there, we can then just pick operating points and run them in the continuous mode. So this is a development or process development which has shown to be very successful, and it's something that can be actually done in very short time.
Ravishankar, do you have anything to say on this?
Yeah, I agree with Britta, what she said, and in terms of the moving from the batch to the continuous. Yes, I agree with her. And I don't have more to add.
Our next question, how do biosimilars manufacturers compete and provide a cost saving relative to reference products? All right,
I'll take this. As I said, most part of my presentation I talked about the cost in manufacturing. How do we do how do you optimize the cost? So what's become very clear is for biosimilars manufacturers, we recognize that we have to be competitive and provide very cost saving or the cost effective drugs, and that's the imperative for any biosimilar manufacturer. How do we do that? There are many ways. One, I could in a very generalized and way of saying is, one is to have a better improved expression systems which can give very high titers of the cell lines that we are using. Adapting better technologies, some of that we talked about in my presentation and in Britta's presentation. reduce the capacity requirement, because I already said that capacity also brings in the cost, because of if we are having a fairly large facility, the cost of operation is also very high. So we need to work on what exactly is the capacity required, and is there a way of reducing the capacity to bring in the competitiveness. And we should also have better methods in terms of the testing of the product and also monitoring of the products. And if possible to have a continuous monitoring of the entire process from start to the end. If you have that, that will bring in efficiency, and efficiency brings in making it more and more yields, and that brings down the cost of the product. So that's one way of saying that any biosimilar manufacturers we can develop and complete in the market with a low cost of the drugs, giving it to the patients. That's my way of looking and saying.
Thank you for that. Our next question is, when does it make sense to consider a continuous chromatography?
So a continuous process can make sense whenever basically you're seeking to intensify a chromatography step. Intensify can mean to reduce the column volume, for example, increase the binding capacity, or also shorten processing time. Typically, bind dilute chromatographies are most efficient when considering continuous chromatography, especially when we have high-priced resin, so talking about protein A chromatography solvents, for example, or some expensive custom-made solvents, for example. This is when continuous chromatography can be considered. Especially when we are working with high-titer feedstocks, then the continuous chrome gives us the most potential compared to the batch process.
Thank you, and unfortunately that's all the questions we have time for, but I'd like to thank you for your answers. And before I finish the webinar, I'd like to ask our presenters, Ravishankar Kasturi and Britta Manser, if they have any closing remarks.
Not from my end, Stephen.
Romy, thank you very much for joining the session today and the interesting questions. I'm looking forward to engaging in further discussions about continuous bioprocessing with you to drive intensification. Thank you very much.
Well, thank you both for that, and I would like to take this opportunity to once again thank our presenters, Ravishankar Kasturi and Britta Manser, for sharing their knowledge with us. I would like to remind our audience that they can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. Thank you, everyone. Have a great day.
Thank you.